In today's lesson, we are going to look at where all of this ATP and NADPH are going. So it is the Calvin cycle that we're looking at, and this is also called the dark cycle or dark reactions because light isn't necessary in the Calvin cycle. So it is light independent, so there's no photons of light necessary. This can be happening at night when there is no sunlight available. Also called the Calvin cycle, and there are 11 enzyme catalyzed reactions, but there's only a couple that we're going to look at. So it's not like the citric acid cycle or even glycolysis where we really focus in on what's happening at each step. We're just looking at an overall production and usage of ATP and NADPH. It starts off with carbon dioxide, and we're ending up ultimately with glucose being produced. A lot of ATP and a lot of NADPH are required in the Calvin cycle, and we get to see now why so much ATP and NADPH were being produced in the light reactions. So in the first phase, we have carbon fixation. This is where we have the conversion of inorganic carbon in the form of carbon dioxide to an organic form of carbon. And you can think of organic carbon as any kind of carbon that has hydrogen bound to it. So in this case, we have these three carbons, and we can imagine there's hydrogens coming off of them. Whereas in carbon dioxide, it's just carbon and oxygen, so it's an inorganic form. So that's a little trick to determine if something's organic or inorganic from the biology person. Your chemistry teacher might disagree. So we have our ribulose 1,5 bisphosphate. So ribulose, 1 carbon, 5th carbon, phosphate. So there's bisphosphate because there's 2. So it's a 5 carbon, and it's joining with a 1 carbon, carbon dioxide. The main enzyme that we focus on is the enzyme Rubisco, and this is the only one I ask you to memorize. And it's really important because it actually does a lot of jobs, but we're only looking at this job where it brings together ribulose 1,5 bisphosphate with carbon dioxide. And this is giving us a 6 carbon molecule. This 6 carbon molecule splits, you can imagine right here, to give us 2 identical 3 carbon molecules of 3 phosphoglycerate, 3 PGA. So again, we go from a 5 plus 1 carbon molecule to give us a 6 carbon molecule with a phosphate on each end. That's going to split right in the middle to give us 2 identical molecules of 3 PGA. And what's interesting is that every carbon atom in every cell of virtually every living thing has actually taken part in this reaction. So all of the carbon of your body has taken part in this reaction. And it's also called, or also known as, C3 metabolism. And that's because we are ultimately ending up with 3 carbons at the end. The next phase is the reduction phase, and we have an ATP molecule being used and NADPH, so there's energy being used in this stage. And this is where we go from the 3 carbon PGA molecule, and a phosphate is being added so that we have phosphorylation happening. And we also have the reduction of that molecule where and the loss of a phosphate group. And it is producing glyceraldehyde 3-phosphate. So this is G3P, which you may remember from glycolysis. The last stage is called the regeneration stage. And this is where we left off, G3P. So we're going to have some G3P leaving the cycle to produce glucose, but we also have some G3P staying in the cycle to regenerate this ribulose 1,5-bisphosphate so that it can continue in the cycle. So it looks a lot like the citric acid cycle, where we have some of the reactants at the beginning end up being regenerated in the cycle. And that's so it can continue on with the Calvin cycle, producing more and more G3P. So let's just take a little peek at this cycle to get a little bit more familiar with it before we move on to looking at more into the numbers. So here this says we have 3 carbon dioxide. So we may have figured that out yesterday with our play clay lesson. So we have 3 carbon dioxide coming in. And so that's going to give us essentially 3 of everything throughout. So that's going to end up giving us reacting with 3 ribulose 1,5-bisphosphate. So 3 with 3 are going to give us 3 of this 6 carbon molecule here. And then again, that splits. So if we had 3 and then that splits, each of them split in half, then we're ending up with 6 of our 3 carbon molecules, so the 3 PGA. And then we have some reactions happening. And again, we're not going to focus on that this year. So we just see that we have, again, so we have 6 molecules happening. So we have 6 reactions happening. So we're going to have 6 ATP being used to phosphorylate those. We're going to have 6 NADPH coming in there. So that's the reduction stage. And then from those 6 molecules, we're only getting 1 G3P leaving the cycle to help produce glucose. So the other 5, they are going off to help regenerate the ribulose 1,5-bisphosphate. So that's a lot going on just to produce 1 G3P molecule. So if you look, we notice that there's 3 carbons in this G3P that will be exiting the cycle. And if we stop and think, you'd be like, miss, glucose has 6 carbon. So what's happening? Well, we can think almost from here to produce glucose, you're going to get the reverse of glycolysis happening. So we're going to end up actually having 2 of these molecules have to be produced to make 1 glucose. So I'll say that again. This is only a 3 carbon molecule, so 2 of these have to come together to produce 1 glucose. So that means we actually have to get 2 turns of this cycle in order to get 1 glucose molecule. So looking at this diagram, it is the one that you would find in your textbook. We see, again, we have 3 carbon dioxide coming in for phase 1, where we have the fixation of carbon dioxide. And it is reacting with 3 ribulose 1,5-bisphosphate coming in, and you don't see it here, but it's producing 3 6-carbon molecules. And each of those are going to break in half, where we would then end up with 6 3-carbon molecules. And that's 3-phosphoglycerate. Some reactions occur, which we don't need to worry about, where we're getting 6 ATP being used because there's 6 3-carbon molecules, and we're using up 6 NADPH, and this is during the reduction stage, to produce 6 G3P. Now, one of these is going to go off and combine with another one to produce glucose. So again, this is like the reverse of the beginning of glycolysis, these stages. 5 of the G3P remain in the Calvin cycle. Again, we are using up 3 ATP, and that's giving us 3 ribulose 1,5-bisphosphate, so it can continue in the cycle. So again, to make 1 G3 G3P molecule, we're using 3 carbon dioxide and 3 Ruby P. We're using up 6 ATP, 6 NADH, and then another 3 ATP. So a total of 9 ATP and 6 NADPH are used to give us 1 G3P. To get 1 glucose molecule, we have to do the cycle twice. So that would end up needing 18 ATP, so that's 9 in total times 2, and 12 NADPH being needed just to produce 1 glucose molecule. So a lot of energy being used. Now, if we follow G3P, which takes 1 turn of the Calvin cycle to produce just 1 G3P, it's going to take 3 carbon dioxide, again, to produce 1 G3P molecule. We're going to end up with glucose. Again, we're going to need 2 cycles of the Calvin cycle to get 1 glucose molecule. And again, this is essentially done by reversing the first half of glycolysis. But that's not the only thing that can be produced from G3P. G3P can also be used to generate fatty acids or amino acids. We can also get different types of sugars. And again, that's following the production of glucose and then the conversion of glucose into storage molecules. So that's the Calvin cycle. So we get to see the whole purpose of creating all of the ATP and NADPH.